Welcome everybody. Great to see everybody out here. Well, it's really, I'm Bob Goldberg. For those that don't know me, I'm Vice President, Executive Director of the Knight Campus. It's a real pleasure and really exciting to introduce today our 2024 Distinguished Lecturer, someone who's been a good friend and scientific collaborator for, sorry to say, Dave, going on 20 some years now. Dr. David Mooney is the Robert Pinkas Family Professor of Bioengineering at Harvard School of Engineering and Applied Sciences. And he's a core faculty member, in fact, one of the inaugural people in the Wyss Institute at Harvard University. Dr. Mooney is an internationally recognized leader in bioengineering, whose contributions have shaped our understanding of biomaterials, mechanotransduction, drug delivery, tissue engineering, and immunoengineering. His accolades are many. He's a member of the National Academy of Inventors, the National Academy of Medicine, and the National Academy of Engineering. That's a very rare and impressive set of accomplishments. And this year he received the prestigious American Institute for Medical and Biological Engineering Pierre Galletti Award, which honors his national leadership and outstanding service to biomaterials and tissue engineering, as well as his dedication to advancing the culture of medical and biological engineering communities. With over 400 published articles and multiple patents, many of which have been commercialized into successful products, Dr. Mooney is one of the most highly cited researchers in the field. His groundbreaking work includes pioneering research on how cells sense and respond to mechanical stimuli in their environments, laying the foundation for innovations in regenerative medicine and therapeutic technologies. He and his lab have also developed implantable biomaterial-mediated cancer vaccines to stimulate immune responses against cancer cells. So yeah, he's done some pretty cool stuff that you're going to hear about today. Dr. Mooney is also an active voice in the professional community, serving on industry and government boards, contributing to major engineering societies, and advising as an editorial board member for multiple leading journals. He's been an inaugural member of the Knight Campus External Advisory Board group since 2018, and we meet tomorrow. And he's offered his expertise on really a critical time during the growth and expansion of the Knight Campus. Dave's experience and knowledge have been invaluable contributors to the establishment of many of our programs, and he's personally been an outstanding sounding board for myself and our leadership team as we make decisions that impact the future of the Knight Campus. And so we're just thrilled that he agreed. Thank you, Dave, for coming a day early and being our 2024 Knight Campus Distinguished Lecturer. Please all join me in welcoming Dr. David Mooney.
I want to start by thanking Dr. Goldberg for the invitation and for the introduction. It's really just an honor to be here today to visit the Knight Campus. It's actually been thrilling for me today to get a chance to meet with faculty and students and postdoctoral fellows here and just see the tremendous progress that's been made in a very short period of time to create a culture of research and technology and really excellence. I'm really looking forward to the rest of the visit and seeing getting other facets of the advances over the last few days, but I'm really enjoying the visit. So thank you very much. I feel like I have to make a small personal note. Something came up when we were talking before we got started. I think the last time that maybe you and I, Bob, were standing in front of a group of students and postdoctoral fellows was at a Gordon Research Conference a few years ago, where we had basically gotten some T-shirts to kind of commemorate the conference. And instead of just giving them to the students, we thought, well, let's have a soccer game and have the faculty against the students and postdocs, assuming the students would win the soccer game, and we could then give them the T-shirts as awards. It didn't quite go as planned. And shockingly, the faculty actually won. And so I was forced to give the T-shirts to the faculty, people like Bob, who obviously played an integral role in that victory. And when that evening we were standing in front of the students, we actually did karaoke and saying, "We are the champions and you are the losers," to all the PhD students and postdocs. I still have actually photos of that, no video, thank God. Okay, so I'm going to talk today about one facet of the research in our lab. And what you're looking at here is T cells in yellow attaching to microparticles that are colorized red here. And we're very interested in developing immunotherapies in which T cells can recognize and destroy cancerous cells and tissues in the body. I'm going to have three kind of short stories to illustrate some of the potential here. The first one will be in relation to developing materials such as the one shown here that we can use to manufacture T cells. The second will be materials we can use to generate T cells in the body via therapeutic vaccination. And then the third will be talking about what happens to T cells when they actually get into the tumor microenvironment. Now, before I get started, I do have a few. Okay. My advancer doesn't seem to be working, but I can do it manually. I do have a few acknowledgements to make. Have a variety of different commercial engagements. I will point out the relevant ones as I go through the talk today, but I do want to acknowledge that the work I'll describe has been funded by the Cancer Moonshot Initiative from the NCI and NIA. Also, some of the work I'll describe has been funded by the FDA and also the NSF and NIDIB. Now, the cell type I'm going to talk to you about today is a particular type of T cell called cytotoxic T cells. And the reason that we're talking about these cells today is that these cells circulate throughout the human body, and these are these red cells you're seeing here. When they encounter foreign agents in the body, which might be a microorganism that's invading, or in this case, cancerous cells that have mutations, they have the capacity to recognize these foreign cells and then to destroy them. So basically clear the cancer. Now, one of the things you probably noticed from this video as well that's quite striking is when they do this, they specifically can kill the cancerous cells, but not any of the healthy surrounding cells. So they can be very specific in terms of how they deal with cancer. That's unlike kind of the classic approaches we treat cancer with, things like chemotherapy and radiation. So this has led to a lot of enthusiasm and excitement about the idea of trying to orchestrate these T cells' response to cancer to get more effective cancer killing. Because obviously it doesn't work this way all the time, or we wouldn't have patients that have cancer. Now, one of the strategies that's been very popular for the last 10 to 15 years to try to get cancer cells to be highly effective in clearing cancer in the body is to isolate T cells from patients and then to genetically modify them, as shown here in the schematic with this CAR antigen recognition domain. It basically is like a label we put on the T cell so it can now recognize specific types of cancer cells. And then those cells from the patient that have been genetically modified are transferred back into patients. And this is actually an image from one of the first patients that was treated with what they call these CAR T cells. And what you can see from the text on the bottom is it led to really remarkable responses in these patients that otherwise would have had very poor prognosis in terms of survival. So they were remarkably effective in a variety of blood cancers, and the initial clinical success of these led to this exponential growth in interest in clinical trials exploring these cells. So there's about 7 CAR T approved therapies today that are used to treat patients, and there's hundreds that are being developed. Now, our part of this comes in from the manufacturing side. So if you look at how these cells are actually made to be applied to patients, is it starts with isolating T cells from the patients, and one can isolate them from a variety of sources. But then outside of the body, these cells are activated, they're genetically modified if relevant for the particular therapy, and then they're expanded to generate enough cells to treat a patient. And in particular, this last step typically takes weeks or even months in the early stages of these therapies before the cells are then taken back to a patient. Now, there's a variety of limitations in this process. One is that it takes a very long time, tends to be very expensive. And then at the end, the product, the cells that are given to the patients tend to be highly variable in their effectiveness. Sometimes highly effective, sometimes not. So our entryway into this space was from the perspective of whether we could develop technologies that could improve T cell manufacturing and control and specify the activity of these cells so they potentially could be more effective when they're used to treat patients. Now, to understand the system that we put together, just going to give a little bit of biology in terms of how these cells are normally activated and then how this is done in an artificial manner. So normally T cells in the human body, to become activated, they interact with another immune cell called an antigen presenting cell or a dendritic cell. And that's shown schematically here on your left. And these cells present three signals to the T cell. They directly engage what's called the T cell receptor by presenting like a little piece of mutated protein or peptide to the T cell, to the receptor the cell has. It provides co-stimulation with another receptor, and then it also secretes different agents that, for example, might make the T cells proliferate better or survive better. Now, how this has been translated to manufacture T cells for patients is, instead of using a cell, which gets kind of cumbersome because then you have two different cells, and if you're taking these cells from patients, maybe they don't function very well because the patient has a disease. So instead, the industry standard today is to use small polystyrene beads, so basically a rigid fixed substrate to which they attach antibodies that can provide signal 1 and 2 here. So this is really different than how a cell would normally present these cues. And in particular, when a cell presents these to a T cell, they interact physically and end up clustering this information together, forming something called a synapse. That's not possible with these fixed signals on these polystyrene or plastic beads. And in addition, the cells in the body normally secrete agents that then act very locally on the T cells. And what's done in this commercial process is you just basically dump cytokine or these signals into the cell culture media. So we wanted to develop a system that could maybe mimic more closely how a cell normally activates T cells. We thought that might be a better way of doing things. And the system we developed is shown here, and it was originally developed by Alex Chung, and then David Zhang is shown their caricatures up on the top. We're starting with microparticles that have a similar dimension in the lateral sense of a cell, but we make them much longer than a cell normally would be so they can engage many cells. This is what these look like under scanning EM. You can see they're about 80 microns long. These microparticles have long aligned nanopores where we can park cargoes like these cytokines that are normally secreted by the antigen presenting cells. And then they can slowly release them. So you can see here's some release kinetics of a few of the relevant agents. So they can release them continuously for a couple of weeks, kind of mimicking this function of a cell. We coat these particles with supported lipid bilayer. And this is just a FRET image showing you that if we basically bleach out the green fluorophore and the lipids, it actually comes back, meaning that the lipids are mobile and can move around. So it's very similar to a cell membrane. And then we conjugate these signals that we're interested in directly to these lipids. Now, unlike a normal cell, where you basically have to take whatever it is that that cell normally presents, here we can precisely define what kind of signals and the quantity of those signals that get presented to the cells. And then once we have those particles, we can mix them with T cells, here in red, and allow them to engage. Now, this system can be used, and I'll show you some data today, in two contexts. One is what we call polyclonal T cell expansion, which means we present cues that engage with any T cell and activate it and get it to proliferate. And so that's on the left. The other is we do antigen specific. So if we only want certain T cells that can recognize specific aspects of cancer, for example, to expand. And I'll give you some examples of both today. Now, in terms of the first thing we want these cells to do, or these artificial cells to do in terms of activating T cells, getting them to expand, this is one of the first experiments, very simple experiment. We mix these artificial cells with T cells that were taken from human patients. What you see is that over time, the cells form these little balls, and the cells are very actively expanding. And you can see with this artificial system, what we call APCMS, we get about a 3,000-fold expansion of the original number of cells over a short period of time. If we look at the industry standard Dynabeads, we get less than a third that rate of expansion. So we can make cells much more rapidly. This also allows us to make cells more inexpensively. And what the data on the right shows you is that the one metric of the function of these cells, something called their exhaustion, is very low with both, indicating that the quality of the cells is similar, even though we're making a lot of these cells and a lot more quickly. Now, that was important, but what was more important is that we can actually control the function of these cells. For the next series of experiments I'll show you, we've been looking at how to regulate the function of the T cells to hopefully make them more effective. Now, kind of as a little bit of background to this as to why this is a challenge. If you think about classic drugs, like small molecule drugs, Tylenol, things like this, those drugs have a defined chemical structure. When we make them, they're exactly the same thing every time. Now, more complex therapies that we use very frequently today, things like proteins, more complex structure, but again, it has a defined composition, so we know what it is, and it's going to have, we hope, pretty much the same exact function every single time. But a cell, when we use a cell as a therapy, a cell can have at least hundreds, thousands, maybe even greater numbers of different functional states. And some of those functional states might be highly effective, and some of them might not be effective at all. So we really need to get a handle on how to control the function of the cells so they can actually be useful broadly in many, if not all, patients. To give you a little sense of, kind of what we know about this challenge when we first got started, I won't go through a lot of the specifics, but T cells have different states of differentiation, as indicated across the top here, whether you go from naive to memory to effectors. And this was an experiment here on the lower left, done in an animal model, where they took T cells at different states of differentiation, and then they would infuse them into a preclinical model of cancer. And what you can see from these different curves is they had greatly varying efficacy. So depending on kind of the state of the cell, it worked really well or it didn't work very well. Similarly, on the right-hand side is some data basically of patients that were treated with these CAR T cells. And the CAR T cells were characterized before they went into the patients for these exhaustion markers. And what they found is the T cells that went into patients that had more markers of exhaustion were much less effective and led to non-responders or non-responding situations in patients, while those that were less exhausted were much more effective. So we actually know from the clinic and from a variety of animal studies that actually this functional state does really matter. Now, one of the nice features about the system that I just described to you is we have great control over how we put it together. So we can vary the particles, we can vary what we call these mitogenic cues, we can vary the lipids, change some of the biophysics of the way the cells interact, and we can vary basically these T cell stimulatory ligands. And we've actually looked at all of these. I'm just going to show you a few pieces of data where we've varied just one of these things, which is not even the type of T cell ligand, but just the density and the amount of T cell ligand. And then we've looked at a variety of features of the T cells that we manufacture and try to understand the relationship between the two. Now, the approach we took here was very much an unbiased approach. So we basically just created a wide array of different these artificial cells. And then we added them to T cells, and then we basically looked to see what happened. So we didn't try to have a hypothesis here. We just did a very unbiased approach. We then looked at a lot of different metrics of the function of the cells. And then we used a technique that's very common in analysis of gene expression, which is called principal component analysis, that we took all these different things we measured and we kind of collapsed it into 2D. And each one of these dots represents a different manufacturing run of T cells. Now, we found that we had a lot of different outcomes. Another technique that's common in the gene expression area is to do clustering, where you say, okay, these groups are kind of similar to each other, so we're going to treat them all the same. And so when we take this approach, we end up with a number of different clusters. And then if we characterize what these clusters represent, what we find, for example, if we look at how much expansion of the T cells occurred, the cells in these different clusters had vastly different expansion. If we look at this marker for exhaustion, vastly different levels of exhaustion, different levels of differentiation of the T cells, and then you see CD4 to CD8 ratio, which we also know is important as well. So we could take the same starting T cell population, and depending on how we manufacture, to get vastly different outputs in terms of the T cells. Now, a question, of course, is whether this really matters. So to explore this, we used not T cells from healthy patients or healthy donors, as in that experiment. We actually went to T cells from patients that were coming into the clinic for treatment for cancer. And this would be patients that had acute lymphoblastic leukemia or acute lymphocytic lymphoma. And this work was done in collaboration with Kathy Wu and Jerry Ritz at the Dana-Farber, and Nico, one of her postdocs, Sid, and then Kwasi in my lab were integral players in this part of the story. And they basically did the same thing that I just showed you, but based on what we learned, we said, okay, we're going to pick a few conditions and create T cell products. And the key thing here is if we look at expansion to start with, you can see, well, first of all, the D is the Dynabeads, the industry standard. These patient-derived cells don't expand very well with those Dynabeads. With our system, we could get varying levels of expansion depending on the amount of stimulatory cues we place in the material. Too little, you don't get much expansion. Too many of these cues, we actually overstimulate, and then the cells lose functionality. You can see that as well if we do a cell culture killing assay, which basically these curves just indicate kind of how rapidly you lose the killing effectiveness of the cells as you give them more and more cancer cells to kill. Probably the more readily kind of understood data, though, is from an animal model. So this is a standard preclinical model developing these products. And what we do is we take mice that do not have an intact immune system, and then we give them human cancer cells. So it becomes a humanized mouse. And then we give them a human CAR T therapy to see if it can effectively clear the cancer. And the cells are actually labeled. So in these images, if it becomes brighter and brighter, it means the cancer cells are multiplying and basically overwhelming the animal. If you don't treat, all the animals succumb within a couple of weeks. And what was really striking here is we could take the same starting T cell population and we could stimulate them in a way that we got highly effective therapeutic cells or highly ineffective therapeutic cells, just depending on how we manipulated the cells. Now, so the first thing this told us is we can indeed get a high level of control and we can make these cells effective. If we go back a slide, the individual dots you're looking at here, let's say if you pick out the orange, those are cells from an individual patient. So the other thing you can recognize is that patient-to-patient variabilities is extremely high in these cancer patients in terms of the responsiveness of their T cells. So we can, on average, make things much better, but we'd like to not just on average make things better, we'd like to make it possible that each patient's cells could be made to be highly effective. So the way that we're approaching this is to use some AI or machine learning models, where what we've done is taken the data we've generated from all these runs of manufacturing cells, and we've created a model where the input is we say, okay, what are the properties of the starting T cells that we get from the patient? And then we define what we want at the end. What will make a functional T cell at the end? And we've generated a model then that can then tell us exactly how we want to construct our artificial cells so that we get the same functional product at the end each time. And so the model at this point in time works relatively well, about 80% accuracy. And so at this point in time, we need to validate this with much larger sample sizes for many more patients. It looks like we can begin to take properties of patients and then design a specific artificial cell to get effective T cells at the end of the process for them. So this is something that we're pretty excited about. We've shown that not only can we do this with the polyclonal T cell expansion, but if we look at antigen-specific T cell expansion, where this is relevant is if we take T cells, let's say from a patient, and we sequence them, we know what mutations they have, and so what T cells should be effective. We'd like to be able to manufacture, or basically specifically enrich and drive expansion of those antigen-specific cells. So again, with Kathy Wu and Jerry Ritz, we've been looking at this. And the key thing of this studies is we can now go back and compare to a normal antigen presenting cell. So the real thing that gets used in the body and say, are we better or are we worse? And if we look at the enrichment, so at the end of the process, what fraction of the cells are specific, in this case for MART1, a cancer-associated antigen. When we use our system, we get much more enrichment than we do if we use normal antigen presenting cells. We get much more expansion. And actually the plots over here just show you again some of the variability between patients. So each one of these is a different patient outcome. But what you can appreciate, the line connects the same patient that was expanded either with the monocyte-derived dendritic cells or our system. And you can see this artificial system always dramatically outperforms the natural cells. And I won't go through all the specifics here, but if we look at a lot of metrics of the quality of the cells, we also dramatically enhance the quality. So we can, again, manufacture more cells and better cells when we use this artificial system. Now, the last piece of data that I'll show you related to this part of the story is sometimes you deliver the CAR T cells to patient. and they're not very effective. And so we became interested whether the same idea we could use to actually rescue a failing therapy. So could we actually inject these microparticles into, in this case, an animal model to start with, in a situation where the therapy is already failing? So in essence, boost an already existing T cell therapy. And so this work was done actually by David and Josh Brockman, who was a postdoctoral fellow in the laboratory. And we went back to the same animal model that I showed you before. Here we purposely used a dose of CAR T cells that would not be highly effective. So you can see that basically they're kind of petering out over time. But if we use the optimized artificial cell and inject it under the skin of these animals, we can now rescue this failing product and turn it into an effective therapy that can then prolong the lifespan of these animals. So I'm going to stop here with this part of the story. And hopefully what I've been able to give you a sense of is how we have defined an artificial cell that we can have a variety of inputs. And based on this, we can then manufacture cells in a way that saves time, provides more control, and gives more function. We've been doing this with T cells. More recently, we've been looking at a variety of other inputs beyond the ones I've showed you today. And we've also shown that this is relevant to expansion of progenitor cells and NK cells. And some of you may be very familiar with NK cell cancer immunotherapy that's also under widespread exploration right now. Now, I was asked to talk a little bit about the translation of these types of things. This was a technology that we felt was ready to move forward towards application in humans. And so we did form a company called Imulus around this technology a couple of years ago. I founded this company, or co-founded it, along with Alex Chung, who was the first person to develop this technology in the lab. And then Omar Ali, who had been a previous student in the lab, who I had founded a company with a few years previously, and he'd gone off to the venture capital world. But we basically, I gave him a call and brought him back to start up this company. And so the three of us were co-founders. We founded Imulus. And then as we were actually doing the initial round of fundraising, we've been talking to Lyle, which is a cell therapy company that some of you may have heard of. And Lyle was going to be an investor, and then they decided they actually just wanted to buy Imulus. So they went ahead and purchased Imulus, and we decided that actually would be a good plan because it would allow us to move much more quickly towards translation because they already had the resources in place. We actually recruited Eileen Lee, who you'll see in a minute from the next story, a very talented previous PhD student to join the team at Lyle. Lyle has been moving this technology forward. They filed an IND to get approval to start clinical trials. And that IND was recently granted. So they will start treating patients later this year or in Q1 of next year with their next generation CAR T therapy that's manufactured using this material system. So we're really excited to see that progress. And, that'll be the real test to how effective this technology really will be. So, about a year from now, I can probably give you an update on that. Now, I did want to make a couple of comments as well about this kind of process of starting a company. And a couple of things I might mention. One is, most startups do fail. And so I think a key thing, at least from my perspective now, after going through this a few times, that I've learned is if you're going to take a lot of time and do something like this, you want to do it with somebody that you really like to be with. So for me, what was thrilling was to be able to continue to work with Alex and Omar, two people that I basically love, and really be able to spend a lot of time with. And even at the end of the day, if we take our shot and it's not effective, at least I'll have spent a lot of time with people that I enjoy being with. So that would be one piece of advice for those of you thinking about starting something up. Do it with people that you like. I think that's actually really key. Okay, I'm going to change gears now and talk about a different way we can think about trying to generate T cell therapies against cancer. And this is to directly generate T cells in the body using vaccination. And we developed a number of years ago, now you can see about 2009, we developed an approach to try to vaccinate using a biomaterial scaffold to generate T cell responses. And the basic idea is shown schematically here. We designed a biomaterial that we'd place in the body and in essence would create a new home for immune cells. And the first thing we would do is we would recruit antigen presenting cells, those cells I was talking about in the last part, to come and basically live inside this material. And the idea is if they're living in the material, we now create a physical separation from the rest of the body And now we can give them cues, what I'm generically calling programming cues, to direct them in the way that we desire. At the same time, we're giving them antigens. And then we direct those cells after they've lived basically in this little cell factory for a little while in the body to leave, go to the draining lymph node and talk to T cells and B cells and generate the kind of responses that I was just talking about. But in this case, it would all be in the body instead. Now, we have been working on this concept for about 15 years. As Bob had mentioned in the introduction, the first generation device, which was a small implantable device, you see it here, held between two forceps, about the size of a baby aspirin tablet. We called it WDVAX. It did go to a phase 1 clinical trial in stage 4 melanoma patients at the Dana-Farber. The trial was run by Steve Hody, and this is Mary Gooding, one of the first patients that was treated with these little implantable biodegradable polymer vaccines. And it was supported by many other people. When you put a clinical trial together like this, and this was an academic trial, it was a huge team, a huge effort to put this together. The general technology was licensed then to Novartis in part, and then we also started up a company out of the Wyss called Atavari with, again, a team of people I was working closely with there. And they've been basically developing this technology to try to push it down the road to hopefully get it something to a point where we could use it to routinely treat patients. What I'd like to do instead of talking about the first generation device, though, is talk about the next gen device, which we've been developing now in the lab for about the last 10 years or so. And two things we learned from our initial clinical trial is one, it was actually pretty cumbersome in that you had to make a small incision in the patient's skin and then place it. That works great in Dana-Farber or University of Oregon Health Center. It doesn't work great in community clinics. So we needed something that could be needle injectable. The other limitation was we couldn't actually make the device until we had a biopsy of the patient's tumor, because then we'd actually freeze dry that, and that would be the antigen we put in the device to basically train the immune system. So we'd get the biopsy, it would take us a few weeks to make the vaccine, then we'd have to implant it. So pretty awkward. So we decided we needed to have a system that would have the attributes shown here. First of all, we decided to go to microparticles that could be readily injected via needle. And then the other thing we wanted to do was to be able to pre-manufacture all of it except the antigen and have it basically be able to sit around, lyophilized, sterile. And then when we had the antigen, we could simply add the antigen, combine it, and then 15 minutes later, go ahead and inject it to a patient. Now, there's some more intricacies of this from a scientific perspective. If we're making microparticles and we're mixing microparticles together, we can have different surface chemistries. We can have a lot of control over basically how quickly or how slowly things get released, which could be very important in terms of tuning the immune response, but I won't really get into that today. The strategy we took to actually make this work was actually the work of Eileen Lee, who I showed you her picture a minute or two ago, and Jae-Yoon Kim, who was a postdoc at the time. And it was basically using these same microparticles that I described as the base for the last part of the story. Have these long aligned nanopores we can use for sustained delivery. Now, the key feature here is that the particles are small enough that we can inject via needle with a saline carrier. But once they're in the body, the saline dissipates in the surrounding tissue. The particles collapse. And in essence, we self-assemble a little home for the cells inside the body. So we allow the home to form once the particles are in the body instead of outside the body. Now, I'm going to actually skip this slide, basically just show that we can orchestrate the immune cells coming to the device, getting activated, and moving to the lymph node. Instead, I'll just show you a couple pieces of data indicating that this actually can work pretty well. So when we use this as a vaccination system, we can look for antibody responses like the COVID vaccines we all got over the last few years. One of the striking things we found about the system, and there's a couple different flavors of it here, but in general, with a single vaccination with the strategy, we can get robust titers that go for well out over a year. So instead of needing frequent boosting, we can basically do single shot vaccinations that lead to really durable responses. And in terms of the T cell responses, which I kind of motivated this with, if we look at a variety of different types of antigen, intact proteins, small peptides, or what are called neoantigens, which represent mutations in basically patients' cancer, If we use those as antigens, in all cases, if we look at the number of activated T cells, which is on the y-axis of these lower graphs, you can see in the VP, which is kind of the optimal vaccine, we generate large numbers. So we can indeed generate antigen-specific T cell populations with this strategy in animal models. If we look at cancer models, where now in this case, again, the animals have, in this case, solid tumors, we vaccinate, and what you can appreciate is, the tumors have a certain size. If we don't do anything to control, they continue to grow exponentially. But of the VP, the optimal cancer, or the optimal vaccine, we get complete regression, and those animals go on to be long-term survivors. Now, we've also shown that these immune responses that we generate have a really robust memory. So, in this case, 200 days after the animals were cured of their cancer, we go back and reintroduce cancer. And we find that actually all the animals that survived and had complete regression of the tumors the first time are able to completely destroy the cancerous cells the second time. So we generate robust responses that has long-term memory with this strategy. Now, we've been exploring this system in a variety of different contexts. One is this idea that we identify antigens that are specific for each patient, and then we make a vaccine for each patient. And that's a big theme in cancer immunotherapy and something we're actively pursuing. But what would be really nice is if we could have a vaccine similar to like the COVID vaccine, that you could give the same vaccine to everybody, or at least a large population, and have it be highly effective. So to pursue this idea, one thing we've been doing is looking at common mechanisms by which cancer cells escape T cell immunosurveillance. And there's actually been a lot of work done to understand this. So one particular way this happens is that cancer cells are stressed in the body. They have a lot of stress on them. And when they're stressed, they actually express genes that lead to proteins on their surface that normally tell T cells to come along and kill them and take them out. But they've evolved a way of avoiding this. So even though they initially express these markers, which are shown here in these red and blue on this cancer cell on the top, they actually have developed mechanisms to shed them. So they actually just cut them all off. So now the T cell or natural killer cell can't recognize them anymore. And actually, once these are shed, they can still bind to the immune cells and actually downregulate the immune cells. So what we proposed in collaboration with Kai Witcher-Fenning a few years ago was maybe we could vaccinate against this escape approach. Now, this is what these proteins look like on the surface of the cell. And the site where they actually get cut by the cancer cells to basically avoid detection is down here, while the site that the cells recognize is up there. So they're physically removed from each other. So we proposed that we could use our vaccine to generate antibodies that would bind at the site of proteolytic shedding, prevent the cancer cells from being able to lose it, and now they stay marked for destruction from T cells. So that was the concept. We're able to successfully demonstrate this, where we would vaccinate, and this just shows schematically summarizing a lot of data. This actually led to maintenance of these markers on the cells, much less of these proteins in solution to downregulate the immune cells away from the tumor. This led to enhanced function of these antigen-presenting cells, and we showed that both T cell as well as natural killer cell functionality was dramatically enhanced. Now, to get a sense of how this might function down the road in the clinic, because this is still a ways away, we went to a couple models of metastatic cancer, which the metastases of cancer is what typically kills people, not the primary tumor. So to model this, in this model, which is basically, this is a melanoma model, we allow tumors to form in animals, but then we surgically resect them. So we basically remove the primary tumor just like a patient would get surgery. What happens though over time is that some of these cancer cells reappear, make it to the lungs, create metastases that will kill the animals. And what you can see on the data on the right is if we have what we call this MIC-B vaccine, we can dramatically decrease the number of metastases at the lungs at later points in time. And this works for many different kinds of cancer. So here we're looking at actually a breast cancer model, same idea. We resect the primary tumor, we look for metastases at later time points, and vaccination can dramatically reduce the metastases, in this case, to other organs. So this general idea of being able to target kind of broad escape mechanisms might be really interesting in addition to targeting the specific mutations in cancer. Now, all this together led us to begin to wonder a little bit about why the system works so well. It looks really potent, really robust responses. And so in the last couple of years, we've been digging a little deeper to try to understand why this works. We thought that part of it was probably just a numbers game, in that we're getting basically more immune cells trafficking to our little factories in the body and then getting processed. And indeed, we do see that. So if we look at the draining lymph node downstream of where we vaccinate, and this is some work of Alex Najibi, we see that with what we call the MPS vaccine, that lymph node actually expands and gets much larger, which some of you may follow your COVID vaccine. Some of you may have noticed your lymph nodes expanding because that's actually the same thing that happened there. But here we're getting a significant expansion and a durable expansion that actually lasts out for a couple of months. Now, if we look at the cellular composition of those lymph nodes, we see we have a lot more antigen presenting cells, many more T cells, many more B cells. So that all supports the idea that we're just kind of ramping up the immune system in a general way, and it's a numbers game. But as part of this study, we also decided to do some single cell sequencing of the cells in the lymph nodes to kind of look at what the gene expression of all these different cells. And I won't go through all the details of this, but basically this was in collaboration actually with Shannon Turley at Genentech. And what we found is if we look at a variety of cells that we know are really important in generating immune responses and just looked at the number of genes that were significantly upregulated or downregulated, you see we're getting these really big changes in gene expression in these immune cell populations. So this suggests that not only is it a numbers game, it's also a quality game, that we're fundamentally changing the function of the cells in the lymph nodes that are responsible for generating these immune responses. Just to give you a little bit of sense of this, if we look at basically inflammatory monocytes, which are important antigen-presenting cells, we see that there's many more of them in the lymph nodes with the MPS vaccine than a naive animal or one that gets a traditional bolus vaccine, kind of the same kind of vaccine as we typically get, just antigen and adjuvant in solution. If we look using this kind of analysis for gene expression at the distribution of the cells, we see that we can break these monocytes into kind of three flavors based on their gene expression: the red ones, the green ones, and the blue ones. And if we look at the MPS vaccine, we see we have a lot of the cells up in this cluster 0 that are not present in the naive and the bolus. And if we look at the gene expression that represents this grouping, we see it relates to things like antigen processing, interferon gamma response, inflammatory response, MHC2. So really supporting this idea that we're fundamentally changing the biology of these cells in the lymph nodes and not just having more of them, but also making them much better at actually presenting antigen and generating T cell responses. Okay, I think actually when I realized there's more kind of a community type of talk, I went a little slower than usual and tried to explain kind of more of the background. So I'm actually going to skip the last part of my talk right now, which related to, okay, what happens when T cells actually get into tumors and how do the physical properties of those tumors actually impact the behavior cells. So I apologize, I'm going to skip right by all of these slides, which are some fun videos, but we will skip that and kind of go right to the end, which is what I hope I've been able to give you a little flavor, is how as engineers, we may be able to play a really important role in cancer immunotherapy. If we think about T cells alone and kind of the T cell life cycle is shown here, we're working on multiple different facets of this. So in some settings, patients don't have enough T cells, period. They don't have enough T cells. So we've been developing technologies that mimic key aspects of the microenvironment of the bone marrow, where the progenitors for T cells are created, and using those as basically injectable devices to enhance the number of T cells in experimental animals, and we hope in a few years in patients. These progenitors actually go to an organ called the thymus, where they differentiate and become functional T cells. We're using basically pluripotent stem cell technology to now try to engineer artificial thymus to enhance T cell differentiation and what's called selection processes to make them better T cells. I told you a little bit about how we're using biomaterials as therapeutic vaccines and how we're basically developing materials to enhance T cell manufacturing. And then the last part of the talk I didn't really go into is we're using material science to try to understand what happens to T cells when they get into the tumor and how to then actually make them function better. So I will wrap up here. And I think at this point in time, we move to a Q&A.
Have some microphones that are going around the room. I'm sure there's lots of questions. Natasha already has one. Go ahead.
Yeah, I'm trying to know, when you ramp up the dendritic B and T cells like that with the cancer vaccination approach, are you seeing any unintended consequences with autoimmune diseases?
Yeah, so in the early work with WDVax that went to the phase 1 trial, we do see some loss of melanocytes at the site of infection, which indicates we're generating a robust response basically against melanin. We don't have any, there's no indication that there's a negative outcome of that. And actually the clinicians looked at that as being a positive sign, indicating that we're generating a robust response. But if that goes too far, then we certainly would be concerned. One of the limitations of that strategy is we're using a lysate of the patient's tumor. So we're basically presenting lots of things, some of which are antigens we'd like to present, and then a lot of things that are just basically normal parts of the cell. So that's one of the appeals of having more defined antigens like the neoantigens, and also targeting these escape mechanisms is versus kind of having this gamish of everything.
Dave, I saw the first generation biomaterial vaccine many years ago. So it's amazing to see how much progress you guys have made. We've got some young faculty in the audience. You started your career kind of as a technology developer in sort of the tissue engineer biomaterial space. How did you make the decision to transition part of your lab effort into the cancer world?
Yeah, I must say, somewhat accidentally. So we were in the regenerative medicine space doing a lot of work with stem cells, which we still do. And we had this concept that maybe we could move a lot of stem cell biology into the body, and we could have materials that could control trafficking of stem cells and differentiation of stem cells. And started looking around and realizing that it was going to be we didn't know enough about stem cell biology to make that happen. So we started looking around and realized, well, immune cells, we actually understand how to make them traffic. So it really started as a proof of principle for this idea of creating cell factories inside the body that could control cell trafficking. But then we started pursuing it, and we actually found that it was generated really potent and robust immune responses. And we realized through talking to some of our now clinical collaborators that was just something we had to pursue. We felt that it looked like it was promising enough that it's something that we really needed to pursue. So we started jumping into that. Actually, I went back and audited immunology courses so I could learn something about immunology because I knew very little about it. And then the lab kind of has done a turn where it's about a third of the people in the lab now work in this general space.
Yeah, amazing. Other questions? Danielle.
Hi, Dave. Excuse me. Really beautiful work. Thank you for sharing. It was some really nice stories. I wanted to ask about the materials that you're using, and I don't think that I heard much beyond the lipid bilayer.
Yep.
So I think probably these are PLGA based materials, or are they different?
Yeah, actually, I apologize. I think I was keeping things pretty general today. The microparticles are actually mesoporous silica. So this is silica that is amorphous. So it dissolves, which is a key feature here. So it will, when we utilize it in the context of T cell manufacturing, it completely dissolves in about 10 to 14 days, which also provides another practical advantage is at the end of the manufacturing, we don't have to have a separation step for like they do with Dynabeads. Here it's just gone. In vivo, we design it so those particles last about 28 days so that we have multiple cycles of cells being recruited to the device, being manipulated, and then trafficking to the lymph nodes.
Great. If I can follow up, that's okay. That's really cool. And I was curious, beyond the degradation and profile, like, is there a function of, and beyond obviously grabbing onto the antigens, there's an absorption step, I think.
Yes.
That's critical for that. What are the underlying design principles? Do you think that this could be translated to other materials? And what is the role of the material besides adsorption, do you think that it's playing a role in the immune response?
Yeah. So a bunch of things mixed in there. Yeah. So so these are nanoporous. They have long aligned nanopores, and we can control the size of those pores, which does two things. One, it creates basically a barrier for diffusion, and we can then slow down diffusion by making them smaller and smaller pores, so we can have some manipulation. We also alter the surface area available for absorption to happen. And we purposely went to an absorption process here so we could get rapid loading of antigen basically at time of application. So the nanoporosity is a really important feature here. The other really nice feature why we use, in part why we use this mesoporous silica, is we have really tight control over the surface chemistry. So we can take a wide variety of different types of agents, cationic, anionic, neutral, hydrophobic, and by controlling the surface chemistry, we can control both the absorption and then the kind of, in essence, the affinity. So the rate of desorption to get varying timescales of release. So the control of the surface chemistry is another really important feature here. Mesoporous silica is by itself somewhat inflammatory, which in this context we think is a good thing. The other kind of part of the answer to the question earlier was the other thing that drove us to immunology is we got tired of fighting inflammation, which inflammation is usually the negative outcome when we're doing regenerative medicine with our materials. And I started thinking, well, where would inflammation actually be a good thing? And in vaccines, it probably has at least a partially beneficial effect. Yeah.
Any students with questions? Sam?
Thank you for that great talk. My question piggybacks off of Danielle's in terms of T cell exhaustion, how your biomaterial is able to recover that exhaustive effect. And in segueing to the stem cell regenerative medicine approach, do you see, is that inflammatory response really the only barrier to trying to take a stem cell and enhance its regenerative capacity, overcoming in some sense the T cell opposite of its exhaustion?
So, okay, so I think the first part of the question related to stem cell exhaustion. So what we found is that we need to precisely tune the strength of the activation cues so that we can activate sufficiently so that the cells will become proliferative, but not drive them into exhaustion. And what that relevant range is depends very significantly on the history of the patient from which we're getting the T cells. Patients that have basically had disease for a long period of time will start with cells that basically are quite exhausted to begin with. And so it's actually very easy to overstimulate those and actually expand, but then get all, even more heavily exhausted dysfunctional cells. So it kind of depends on the starting point for the T cells. And it's part of the reason we think this idea of being able to really easily tune, the stimulatory cues we provide from the material will be really relevant. And then let's see, the second part was
Regarding the regenerative medicine capacity, like in terms of using your techniques to enhance the stimulation response for regenerative medicine, taking like a stem cell and saying, all right, let's enhance this ability for it to regenerate tissue.
Oh, okay. Yeah, so we've not done a lot of work with that. We've done some work with progenitor cells and shown that, for example, we can, if you look at pro T cells, for example, that we can actually have a significant impact by with some of these systems, with some of the same cues in terms of balancing stimulatory cues. They're looked at a lot of more the biophysical cues as well. So we certainly can have a lot of impact on stem cells and progenitor cells and try to control their state. I'd say in general, it's not, in cancer, we still don't know precisely what we want for the kind of the optimal effector cell. And I'd say in regenerative medicine, we probably know even less. So we have the ability to tune, but what we specifically want, I don't think any of us really know exactly yet.
Thank you so much.
So Dave, last I looked, CAR T therapy for a patient can run more than $500,000
Yes.
per patient with a success rate of what currently? How often are they? Is it not successful?
Yeah. So in certain types of blood cancers, you kind of have immediate responses, let's say 80%, but then that drops with time. Yeah. In solid tumors, not so effective at this point in time.
Right. And no money back guarantee, of course, for the patients.
No.
How much do you think with this technology you can bump up that level of patient responsiveness?
Is this being filmed?
I
Oh, no, is this,
no, right?
Okay.
No,
okay,
it is? It is, yes, sorry.
okay.
yes.
I'll give a couple answers to this. So one thing I think is really exciting is, so Lyle, in their new CAR T product, which is targeting a different antigen than what I was showing here, that they're showing actually, their early work shows, looks like it's gonna be very promising against solid tumors, which has been kind of the holy grail for CAR T cells. And what's really striking is it looks like, at least preclinically, they're able to drop the cell dose very substantially. And the cost, in theory, should scale with the cell dose.
Yeah.
So, that should, in theory, make it much less expensive. Let's see, my second part of the answer to this question, I have to think about this for a second. Nothing to do with Lyle or this product, but other things we've done in the past in terms of commercialization. I had entered into certain types of technology development from the perspective that if we could make something, a process for therapy much less expensive, that would have a benefit for the healthcare system and the overall society. At least my perspective today is that the prices of therapies do not depend that heavily on the cost of goods or manufacturing. It instead depends much more on what's considered to be the value it brings to a patient. And so if you are able to make a therapy that's very expensive, make it much less expensive to manufacture, I'm not as convinced that will be passed on to the consumer as versus just being a higher profit margin for whoever makes the therapy. So
At least in the US.
in the US, yes.
Yeah.
So that's the reality as I see it.
Yeah.
So
Another question here.
Thank you for such a good lecture. I'm wondering if you're trying to apply this to replace immunotherapies like chemotherapy for a variety of different cancers, or you really want to supplement it?
yeah, it's interesting. So in certain types of cancer, some immunotherapies, like one checkpoint blockade that I didn't talk at all about today, but it's just been hugely impactful, has moved as a frontline therapy. So it may be able to replace some traditional therapies in certain contexts. It may also be combination. So like preclinically, what we've done and a lot of other groups have done as well is, for example, we've shown that we can make our vaccines and kind of skip the whole idea of identifying antigens, instead by placing the vaccine intra or paratumorally. And then when you give chemotherapy and you start killing cancer cells, that releases a lot of antigen that then becomes the antigen source for the vaccine. So, there's some really interesting ways you might be able to combine the two as well. And maybe you can start to go to lower doses in that kind of setting. So I think at this point in time, it's likely that some immunotherapies will be used in place of traditional therapies. More frequently, they'll probably be used in combination and sometimes very purposefully because there might be some real synergies.
Marvelous talk, Dave, as usual. My question has to do with variability, both patient and tumor, and you touched on this a bit in the first part of your talk, where you described that the T cells with which you start can be variable in terms of their characteristics and their character. A bit more about how you deal with that challenge. And then along the same theme is that the natural history of tumors, as we know, is that over time, they mutate and become variable. So once you train T cells to recognize a given tumor antigen, and the tumor mutates and changes antigenic nature, how does one deal with that, and how robust is a vaccine in terms of being able to recognize this changing motif of tumor antigens?
Yes. Yeah, so actually I'll answer the second one first because I remember it more clearly. No, that's a great question. I mean, cancers evolve, which is you're pointing out, and you may target an antigen and then that antigen is no longer there, but the cancer is still there with different mutations. There's a variety of ways that potentially can get addressed. I'll plug one thing that we've done that I think is pretty neat. So Kwasi Aduberchi, one of the PhD students in the lab, had a really nice idea where he combined adoptive T cell therapy with our vaccination. And the idea was that the adoptive T cells would do a very good job of killing a lot of the cancer cells, debulking a tumor. As those cells were dying and generating a lot of antigen, the vaccine could promote influx of a lot of antigen presenting cells and pick up this variety and get something called antigen spread, which is even though the CAR T cells were only against one antigen, by the time the process was done, we then had T cell populations that could recognize many antigens. And he was able to show that then, for example, if we then introduced cancer cells later that didn't have the antigen that we targeted with the CAR T cells, we actually had an adaptive response that was very effective against them. So things like that. Yeah, dynamic vaccine, and really trying to exploit this idea. And there's a variety of ways that one can try to do it, of antigen spread. So even though you're targeting one thing, try to get the response to basically broaden with time. And that was, again, your first question actually was the heterogeneity. Yeah, in terms of the input, the strategy that we've been pursuing is to try to have a minimal set of characteristics that we quantify from the incoming T cell population that we get. And then say, okay, here's some properties, and try to define what we need to know. And then as I kind of mentioned with the machine learning approach, you say, okay, if this is the input and we want this output, how do we, can we use machine learning to connect those dots? And so we're working on that. The patient numbers we have at this point in time are relatively low, is enough to publish, but not enough. I wouldn't want to go to the clinic with that. But as those data sets get built up, I'm hoping that we actually could start to personalize the manufacturing, which of course does have downsides in terms of costs and in terms of regulatory approaches. But that's one approach I can see to go. The other is if we have a better system, and it's been kind of interesting. We don't have a ton of data, but we've been doing the same thing with NK cells. And we found great variability with NK cell manufacturing when you use feeder cells, which is how they're today manufactured. When we use a variation on the system, we're able to dramatically reduce the donor-to-donor variability in terms of our ability to expand NK cells. So maybe we can actually get systems that, even if it's not exactly the same at the end, at least kind of takes the poor responders and makes them okay.
We've got one last question, Tom.
Thanks very much. Due to the hostility to the tumor microenvironment, it certainly makes sense that a numbers game plays into it.
Yep.
My question is the up and down regulation also possibly a numbers game? In other words, when these cells are amassed in such a quantity and have, next door neighbors that are just like themselves, that might not be the case in biology, in actuality, does that induce the up-down regulation that you saw? Or have you been able to show that if you mass these cells together, does that affect the up-down regulation as opposed to just de novo, they're up and down regulating?
So by up and down regulating, I just want to make sure I'm understanding what aspect you're referring to by the up and down.
Well, when you demonstrated in the bottom right-hand corner of one of the last slides where you said it's not just a numbers game,
Yes.
we're actually showing up and down regulation.
Yes.
Is there a possibility that the up and down regulation is also a numbers game? In other words, when you amalgamate this large population of cells, can that induce the regulation as opposed to it happening on its own for another reason?
Yeah, maybe I'll see if I'm understanding you correctly. How I might think of this is kind of from like a pharmacokinetics perspective. If we look at the area under the curve, and how much correlation do we have to just the general area under the curve for these cell number profiles as versus other elements? Does that kind of get it?
A little bit, but also is the presence of just the mass of cells affecting the regulation?
Oh, okay.
As opposed to something de novo that's happening here.
Sort of positive feedback.
Yeah. Don't know. I mean, we know that we are getting qualitative changes in the individual cells in terms of their gene expression, but our data does not exclude the possibility that some of the cell-cell interactions due to the pure quantities, as you're pointing out, might also be playing some significant role here. Don't know.
All right, we're a little past the hour. I'd like to thank Dr. Mooney again for an outstanding lecture. And so thank you all again for coming. For anybody that didn't get any questions answered, we do have a reception outside, and you can stick around for a little bit.
Yes,
And you can come ask them some questions. Thanks again.